Welcome back. We are now going to finish up our unit on the cell by examining the cell cycle. The cell cycle is the process of growth and division that every cell undergoes that ensures the continuity of life through time. In the last web lecture, we thought about this idea of transmitting information through space, through cell signaling, and now we're going to think about the transmission of information through time, the idea of reproduction, the copying of genetic information, and transmitting it to two daughter cells to make sure that that information is carried on intact through generations. So it's also going to be very important to this essential corollary to reproduction, the idea of growth and development. Cells arise through cell division of pre-existing cells. Cells can only come from other cells that existed before. Observations of newly developing organisms or embryos have confirmed that plants and animals start life as single-celled embryos, so a single fertilized egg that's going to undergo a series of cell divisions to create a multicellular organism. So meiosis is going to be the process of cell division that's going to create reproductive cells called gametes, and mitosis is going to produce all of the other cell types in the body, all of the non-reproductive cells, and we're going to call those somatic cells. The ability of organisms to produce more of their own kind best distinguishes living things from non-living matter, as we saw in that list of things that living things do, that distinguishes them from non-living things. And the continuity of life on Earth is based on the reproduction of cells, or cell division. And cell division is also responsible for growth in multicellular organisms. That's how we get bigger. The cells themselves don't keep growing and growing and growing, they divide to create more cells to make larger organisms. So mitosis and meiosis are the process of duplicating genetic material, but these are also accompanied by the process of cytokinesis, the actual separating of the entire cell into two daughter cells. So think of mitosis and meiosis as division of the nucleus. Cytokinesis is the division of the entire cell. During mitosis, the genetic material is copied and divided equally between the two daughter cells. So the daughter cells are going to end up, in the case of mitosis, genetically identical to the parent cell and to each other. So they're going to have exact copies. Each daughter cell gets identical copies of the original DNA. Meiosis, on the other hand, produces cells with half the amount of hereditary material as the parent cell. They're going to divide without duplicating, and the daughter cells are going to be genetically different from each other and genetically different from the parent cell. So how does this creation of genetically identical daughter cells come about? So the basic steps in cellular replication is first copying the DNA. You need to have two identical copies of all of the DNA. Those two copies have to be separated from each other in such a way that each of the daughter cells gets one copy of each chromosome. And then the cytoplasm needs to divide to create two complete cells. So first let's look at what is the stuff involved here. So first of all, what is a chromosome? A chromosome is a single long double helix of DNA. So remember, one molecule, one super long molecule of this DNA polymer that's going to be wrapped around proteins called histones. So recall that DNA encodes all of the cell's genetic information. And when we talk about a gene, we're talking about a section of DNA, but not just any old section of DNA. So a gene is a specific section of DNA that's going to code for a specific messenger RNA, and that messenger RNA is in turn going to code for a specific protein. So a gene is a section of DNA that codes for a single protein. Before mitosis occurs, before the separation of the nuclei, each chromosome is replicated. So each double-stranded DNA copy is called a chromatid. At first, the chromatids are attached all along their entire length by proteins called cohesins. But once mitosis begins, most of those cohesins are cleaved so that the two chromatids separate everywhere except for one spot called the centromere, where they're still attached to each other by these cohesins just at this one spot, and that creates that sort of X-like shape that we are used to seeing. Chromatids attached at the centromere are called sister chromatids. So the two attached sister chromatids are still considered a single chromosome until the point at which they separate into their two separate cells. So let's look at a figure illustrating all of this. So here we have an unreplicated chromosome. This one strand is representing a double helix. We see one teeny little section that is a single gene. And it's going to be wound around these histone proteins, but in a very diffuse way. So this is the very diffuse form of DNA called chromatin. So the next thing that's going to happen is that this DNA is going to become replicated. So here we have two different strands. Each of these strands is representing one of these double helices. So we have two copies of everything, every gene along this entire long DNA molecule. Then, when the cell is getting ready to divide, all of these histones are going to cause the DNA molecule to condense, to scrunch together to form what we see as the visible chromosome that we can actually image and see under a light microscope. It's a DNA condensed around its associated histone proteins, resulting in a compact chromosome that is 10,000 times shorter than its original length. So this is where we get these identifiable kind of X-shaped chromosomes. And here we see a pair of sister chromatids attached together at the centromere by these cohesin proteins. So in 1882, the German anatomist Walter Flemming developed dyes to be able to visualize these chromosomes and observe them during mitosis and cytokinesis. So a lot of this process of the cell cycle has been known for a very long time. And what we observe is that growing cells cycle between two distinct phases. One is a dividing phase called the M phase. M is for mitotic. And in this phase, the chromosomes are condensed into these compact structures that we just saw. A non-dividing phase, which is called interphase. So in interphase, the chromosomes are uncoiled. They form this very diffuse, uncondensed form called chromatin. And during interphase, the cells are growing and preparing for mitosis, or they're just doing their job as cells. They're fulfilling their specialized functions. And this is where cells spend most of their time, in this interphase between cell division, growing, duplicating their organelles, and doing their jobs as cells. So interphase can be divided into three phases. There are two gap phases, so the G1 phase is the first gap, and then the synthesis phase, and then the second gap phase, G2. And so the cell grows during all three of these phases, but the chromosomes are duplicated only during the synthesis phase, during that S phase. Synthesis refers to the duplication of the chromosome, DNA synthesis. So what are these gap phases for? So interphase includes two gap phases during which no DNA synthesis occurs. And here is kind of an illustration of the cell cycle from one mitosis to the next, showing you the proportion of time a typical cell would spend in each of these phases. So G1 is the first gap. It occurs between the mitosis phase and the synthesis phase. And G2 is the second gap occurring between synthesis and mitosis. And during these gap phases, the cells are growing, and they're also replicating their organelles so that when they do undergo cytokinesis, there's a full complement of organelles in each of those daughter cells. They're ready to function normally. So what happens during the M phase, during the mitotic phase? So M phase consists of two distinct events. One is mitosis, the division of the replicated chromosomes, and the other is cytokinesis, the division of the cytoplasm, forming two distinct cells. So mitosis begins when that chromatin condenses to form those identifiable little X-shaped chromosomes. And during mitosis, the two sister chromatids separate to form the independent daughter chromosomes. So one copy of each chromosome goes to each of the two daughter cells, and each daughter cell receives a copy of the genetic information that's contained in each chromosome. So each daughter cell gets a full set of that genetic information. So the mitosis is a continuous process with 5 subphases: prophase, prometaphase, metaphase, anaphase, and telophase. We'll take a close look at each one of those. So let's start with prophase. During prophase, this is when the chromosomes condense and first become visible under a light microscope, and a structure called the spindle apparatus begins to form. So the job of the spindle apparatus is going to be to produce mechanical forces that are going to move the replicated chromosomes into the right position to eventually be divided. So during the early part of mitosis, these chromosomes are going to be kind of jockeyed into the right position. And then the spindle apparatus later in mitosis is going to be responsible for pulling those chromatids apart into the separate daughter cells. The spindle apparatus is made of microtubules. So as we saw when we learned first about microtubules and the cytoskeleton, they're going to form from something called a microtubule organizing center, or MTOC. And the microtubule organizing centers are going to define the 2 poles of the spindle apparatus. So there are going to be 2 of them, and they're going to go to the 2 sides of the nucleus that are eventually going to split apart to form 2 nuclei. In animal cells, these MTOCs are called centrosomes, and each centrosome contains a pair of centrioles. So research has shown that these centrioles are actually not critical to the formation of the spindle, but the functioning centrosome is going to be the center where these microtubules are formed and organized. During prometaphase, this is where the nuclear envelope breaks down, and the microtubules are going to start to grow from these poles inward and attach to the chromosomes at structures called kinetochores. So kinetochores are proteins. They're going to form at the centromere, so they're going to be attached to that area on the chromosome, on the 2 sister chromatids that are joined together by these cohesin proteins, and they're going to form on opposite sides of each chromosome, so that there's going to be 1 kinetochore facing each pole of the mitotic spindle. Microtubules that attach to the chromosomes at these kinetochores are called kinetochore microtubules. And these microtubules are going to push and pull these chromosomes until they reach the middle of the spindle. So what does it mean? What are they actually doing when they push and pull these chromosomes? So remember that microtubules are these long polymer chains of these little monomer units, and so in order to push a chromosome, they assemble more of these monomer units to grow the length of the microtubule. To pull on them, to pull them back, they're going to disassemble some of that polymer chain to make it a little shorter to pull it back toward the centrosome, or the microtubule organizing center. So the job of these microtubules, these kinetochore microtubules, is basically going to be to push and pull and make sure that these chromosomes are in the right location in the middle of the mitotic spindle. During metaphase, this process of positioning the chromosomes is going to be complete. The formation of the mitotic spindle is completed, and the chromosomes are going to be lined up on an imaginary area called the metaphase plate. Remember, there's no actual structure there. This is just an imaginary plane between the 2 spindle poles where all of the chromosomes are going to be lined up. And each chromosome is going to be attached to a kinetochore microtubule coming from each of the opposite poles of the mitotic spindle. So other microtubules are part of the spindle that are going to hold the spindle poles in place. These are astral microtubules that are radiating in the opposite direction. And then there are other microtubules that are part of the mitotic spindle that don't actually attach to chromosomes, but are going to be important in the elongation of the cell for final division. So let's take a look at those early phases of mitosis in diagram form. So here we see interphase, this sort of diffuse form of DNA called chromatin that does not form distinctly visible chromosomes. We've got our centrosomes. This is an animal cell containing these centrioles, and they are duplicated, so there's a pair of them. In prophase, though the DNA is condensing to form chromosomes, this pair of centrosomes are migrating, one to each side of the nucleus. And then in pro-metaphase, we start to see the nuclear envelope start to break down. So we're seeing more and more gaps in the nuclear envelope. The microtubules are starting to contact the chromosomes at these kinetochores, and they're going to start to push and pull them into position. As microtubules from either side connect to those kinetochores, they're going to put mechanical forces on them. That's going to force them into the very center of the mitotic spindle. And here they are in metaphase, all lined up on the metaphase plate with one kinetochore microtubule attached to each chromosome from either side, and they are ready to go into the later stages of mitosis. So the next phase is anaphase. So during anaphase, those cohesins, those proteins that hold the sister chromatids together at the centromere, are going to split. And that's going to allow the sister chromatids to be pulled by the spindle fibers toward the opposite poles of the mitotic spindle. So this is going to create two identical sets of daughter chromosomes, because remember, one microtubule from each pole attached to one of each pair of sister chromatids. Each pole is going to get one complete set. So there are two forces that pull these chromosomes apart. First of all, the kinetochore microtubules are going to be disassembled. So they're going to shrink in length as those monomer units are taken apart, and the polymer decreases in length. But there are also motor proteins that are going to be attached to the microtubules that make up the rest of this mitotic spindle that are going to be pushing the two poles of the cells away from each other. So you can think of these microtubules as overlapping each other to form kind of a circle, and these motor proteins cause them to decrease that overlap and spread apart from each other to make this sort of oblong shape, pulling the poles of the cell away from each other. So during telophase, this is when you start to see a new nuclear envelope forming around each of these sets of chromosomes. And the chromosomes begin to decondense again to form that chromatin. And we consider mitosis complete when the two independent nuclei have formed. Those nuclear envelopes are completed. Cytokinesis typically occurs immediately after mitosis. So the cytoplasm is going to divide to form two daughter cells. There's going to be a division of the organelles in the cytoplasm so that each daughter cell gets all the organelles it needs. And here's a diagram of these late stages of mitosis. Here are the chromosomes attached by the kinetochore to these kinetochore microtubules. Those microtubules are shrinking and pulling the chromosomes toward them. At the same time, these non-kinetochore microtubules that are overlapping each other are using motor proteins to push the poles away from each other, decreasing that overlap and making the cell more oblong. You can see they're kind of overlapping here, and when we get into telophase, they're overlapping very little. Think of them sort of telescoping out to form this elongated cell shape. So in telophase, we see the two nuclear envelopes reforming. The cell has become oblong and ready to divide between the two nuclei. And at this point, cell division cytokinesis begins using sort of a little belt of actin and myosin around the plasma membrane between the two nuclei that's going to start to contract, slide past each other to constrict this ring, and eventually pinching off the two cells, creating two separate cells. Let's take a closer look at this mitotic spindle. The mitotic spindle is a structure that's made of microtubules, and we've seen a lot of ways in which it controls the movement of the chromosomes during mitosis. In animal cells, the assembly of the spindle microtubules begins in the centrosome, the microtubule organizing center, and the centrosome replicates during interphase, forming two centrosomes that migrate to the opposite ends of the cell during prophase and prometaphase. An aster, which is a radial array of short microtubules, extends from each centrosome to sort of anchor it and hold it in place. And when we talk about the spindle as a structure, included in that structure are the centrosomes, the spindle microtubules, and also the asters. So this whole structure that radiates out from this microtubule organizing center is part of what we call the spindle. So again, this is reviewing, but this is gearing up for a deeper understanding of what's actually happening here. So during pro-metaphase, remember that some spindle microtubules are going to attach to the kinetochores of the chromosomes and are going to begin to move the chromosomes. They're going to do that pushing and pulling, getting them into place. And the kinetochores, remember, are protein complexes associated with the centromeres. And at metaphase, the chromosomes are all lined up at the metaphase plate, a plane midway between the spindle's two poles. So let's take a closer look at what happens at this point when all of the chromosomes are lined up on the metaphase plate with their kinetochore microtubules attached to each of the sister chromatids. So in anaphase, those cohesins are cleaved by an enzyme called separase, and the sister chromatids are going to separate and move along the kinetochore microtubules toward opposite ends of the cell. So remember that these microtubules shorten by depolymerizing, and they do this at their kinetochore ends. And how do we know that they do this at the kinetochore ends? Well, we know from experiments like this one, in which the kinetochore microtubules are marked somewhere in the middle, and we can see how this marked section of the microtubules moves as the microtubules are disassembled. So if they're disassembled here from the centrosome end, you would expect this marked section to move progressively toward the centrosome. If they're disassembled here from the kinetochore end, they're going to stay in place because these are coming off here and moving toward this marked area. So what we find when we do this experiment is that those marked regions of the microtubules do not move at all. The chromosomes simply move toward them, indicating that these polymers are being disassembled here from this end. The results of another experiment suggest that motor proteins are also involved in this process. So the proteins on the kinetochores, so this ring here is a ring of motor proteins that's interacting with this microtubule and walking along it. The motor proteins are going to walk along this microtubule during anaphase. So basically, once these motor proteins have passed a section of the microtubule, that's the point at which the microtubule is disassembled into its monomers. This is not only a mechanism for pulling the chromosome over to that pole, but also a trigger for the depolymerization of the microtubule fibers as it goes. And then again, non-kinetochore microtubules from the opposite poles, also using motor proteins, are going to overlap and push against each other using these motor proteins, elongating the cell. At the end of anaphase, duplicate groups of chromosomes have arrived at opposite ends of the elongated cell, and then cytokinesis can begin. During anaphase or telophase, that pinching in can start before those chromosomes have fully separated, and then at that point the spindle is going to disassemble once the chromosomes have reached their final destination at the poles. So let's look at what happens during this process of cytokinesis. Cytokinesis ultimately is going to result in the formation of those two daughter cells, and it's going to happen very differently in plants than it does in animals. So in plants, vesicles from the Golgi apparatus are going to bring sections of membrane. Remember, these little vesicles are composed of cell membrane, and also cell wall components that are going to be contained in the vesicles to the middle of the cell. And they're actually going to use these microtubules from the spindle to arrive at the right location. So remember, motor proteins are generally what's attached to vesicles to move them to the right place. So they're going to go from the Golgi apparatus, they're going to attach to these spindle microtubules. Motor proteins are going to carry them to this metaphase plate between the two poles, and then these vesicles are going to fuse to form a cell plate. So it's going to be composed of cell membrane and cell wall. So when these vesicles fuse, they're going to form a plate that is eventually going to create the cell membrane and the cell wall. So in animals and many other eukaryotes, what we see is this ring of actin and myosin filaments. They're going to slide past each other and contract in the cell membrane. And so this pinching off is going to cause something called a cleavage furrow. So cleavage means splitting. This is going to be kind of an indented area where those actin and myosin filaments are sliding past each other. So this ring is going to shrink and tighten until cell division is complete. So bacteria, remember, have only one single circular chromosome. They're obviously going to do this in a different way, and this different way is known as binary fission. So prokaryotes, bacteria and archaea, are going to use this process of binary fission to divide. So in binary fission, the chromosome replicates. So we're going to have a replication of this circular chromosome, but basically the circular chromosome is going to start as sort of this, what's called a replication bubble, and the two copies are going to sort of spread away from each other as replication continues. So it's going to start at an origin of replication, and then there are going to be two copies in this replication bubble, and these are going to sort of move away from each other. As the two copies of the origin move away from each other, they're going to move to opposite ends of the cell, so that the origin on one copy is going to go to one side, the origin on the other copy is going to go to the other side. This is what's going to pull the two chromosomes to either end of the cell. So here, one copy of the origin is now at each end of the cell, and as replication finishes, everything's going to move toward that origin, and then the cell membrane can pinch off and result in the two daughter cells. So let's take a look at how this process of mitosis might have evolved. So since prokaryotes undergo mitosis in a very different way from eukaryotes, we should think about how these two are connected through evolution by common descent. So because prokaryotes evolved before eukaryotes, most likely the original form of mitosis was binary fission. And there are certain protists that exhibit sort of intermediate forms of mitosis that are not quite like the way in bacteria, not quite like binary fission, and not quite the way eukaryotes do it. And this gives us an idea of a possible evolutionary pathway between the two. So remember, there's no nucleus in the prokaryotic cells, so they rely on this kind of pulling of the chromosome toward the poles to be able to separate the two chromosomes. So they're basically being pulled out toward the cell wall. And so we see in dinoflagellates, which is a eukaryote with a nuclear envelope, a similar kind of system where the two ends of the nucleus are pulled using microtubules toward the different ends of the cell. So this is very different from what we saw in most eukaryotes, where the nuclear envelope disintegrates and reforms. In this case, it splits in a process similar to cytokinesis after the two ends of the nucleus are pulled out toward the outside of the cell, and then they divide. Then in diatoms and some yeasts, again, these are eukaryotes with a nuclear envelope. Now we see a system where still the nuclear envelope doesn't dissolve, it still divides, but now these chromosomes are being pulled apart from inside the nucleus. So this is something more like what we see in eukaryotes being pulled to each side rather than the entire nucleus being pulled toward the different ends of the cell. So again, this is a step more similar to what we do in most eukaryotes until finally we get to the eukaryote state where that nuclear envelope doesn't divide, it just disintegrates and is reformed. So now let's think about the regulation of this cell cycle. What keeps it moving? What makes it happen in the right place and at the right time? So we can see that cell cycle length can vary greatly among different cell types and during different phases of life. When you're actively growing, you've got a lot of cell division going on. When you're a fully mature adult, you don't have quite so much. So most of this variation is due to variation in the length of the G1 phase. And in fact, cells that are dividing really fast all the time essentially just eliminate that G1 phase. They just plow right through it. In contrast, non-dividing cells get permanently stuck in G1 phase, and we give this a special name for non-dividing cells. This arrested state is called the G0 state. Division rate can also vary in response to changing conditions. When there's a lot of nutrients available, for example, cells might divide very, very rapidly in response to those favorable conditions. So all of this variation suggests some form of regulation. Something must be matching the correct response to the correct conditions in some kind of regulation process. And we see the special case of cancer cells that manage to escape the usual controls, the usual regulations on the cell cycle, and just divide completely out of control. So let's take a look at how this happens. So the cell cycle appears to be driven by specific chemical signals present in the cytoplasm. So some evidence for this hypothesis comes from experiments like the ones described in your book, in which cultured mammalian cells at different phases of the cell cycle were fused to form a single cell with two nuclei. But this can also be seen in different kinds of experiments where we simply transplant cytoplasm from cells in different phases of the cell cycle. So in this case, we've got a cell that's in interphase, is injected, microinjected with cytoplasm from a cell that was in M phase, and then a different cell also in interphase, microinjected with cytoplasm from a cell that was also in interphase. And what we see is that the one that was injected with cytoplasm from the M phase cell immediately enters mitosis. So we know that there's some kind of signal that's present in the cytoplasm that's triggering these events. The sequential events of the cell cycle are directed by a distinct cell cycle control system, which we can think of as similar to a clock. Periodic cycling of these different phases. But it's a clock that can be adjusted at different points along the way. So here's a diagram going from G1 through synthesis, through G2, through mitosis. And the cell cycle control system can be regulated by both internal and external controls. So external controls could be things like those growth factors that we were talking about earlier. And the clock also has specific checkpoints where the cell cycle stops until a go-ahead signal is received. So we're going to stop and check things at various points along this cycle, and we're not going to proceed unless the correct conditions are met. The signal that tells the cell to go ahead and go into mitosis is something called an M-phase promoting factor, MPF, also known as a maturation promoting factor, also known as mitosis promoting factor. MPF stands for all of those. So this is present in the cytoplasm of M-phase cells, and it induces mitosis in all eukaryotes. So the MPF is composed of two distinct subunits. One is a protein kinase. So remember that a protein kinase is always a protein that takes a phosphate group from ATP and attaches it to another protein. So this is an enzyme that catalyzes the transfer of a phosphate group from ATP to a target protein. And the other component is a cyclin. So this is a protein that's present in different concentrations throughout the cell cycle. The concentration of MPF cyclin is going to increase throughout interphase. It's going to peak in the mitotic phase, and then it's going to fall off again before it starts to increase through interphase. So this is the cycle, right? It's going to cycle through high and low concentrations. So the MPF protein kinase is a cyclin-dependent kinase, so it's only going to be active when it's bound to the cyclin subunit. So we call this protein kinase, we call it CDK, cyclin-dependent kinase. So when cyclin concentrations are high, MPF can be active. When MPF is active, target proteins are phosphorylated, and that's going to initiate all of these processes that result in mitosis. So here we see a graph of the cyclin concentration here on the y-axis, with time on the x-axis going through the cell cycle. And we can see through interphase, the cyclin concentration is increasing until it peaks just a little bit after the start of the M phase, and then it drops down precipitously to increase again through interphase. So this is the cycle of the cyclin protein. And the CDK subunit has two phosphorylation sites. So we can attach phosphates here and here. Both are phosphorylated after cyclin binds. So when cyclin is bound here, there's going to be another phosphate group attached here to the second phosphorylation site. And the second phosphorylation site is an inhibitory site. So this ensures that this enzyme doesn't start catalyzing its reaction until the proper time. So when the time comes, late in G2 phase, this inhibitory phosphate group is going to be removed, and that's going to activate the kinase and allow the cell to go into mitosis. It's going to lead to the chromosome condensation and this entire cascade of events that leads to cell division. So how is MPF turned off? At the end of the cell cycle, how does it go back into interphase? So MPF deactivation illustrates two key concepts about regulatory systems in cells. One is the negative feedback loop, and we've seen this before in negative feedback inhibition when we're thinking about enzymes, and this is going to occur when a process is slowed or shut down by an abundance of its products. So when the products begin to accumulate, it's going to shut down the whole system. And then the second concept is that destroying specific proteins is a common way to control cell processes. So if something depends on a protein and we just degrade that protein as we go and create new ones, that's going to create an immediate stop if we just stop producing that protein. So the enzyme complex that's activated during anaphase attaches proteins to the cyclin subunit, and those proteins that are attached to the cyclin are going to mark it for destruction. They're going to attract proteases that are going to break down that cyclin. So that's going to lead to the deactivation of MPF. Remember, this isn't active unless a cyclin is attached to it, and that is how the concentration of cyclins decreases as it goes through the M phase and starts again in interphase. So let's take a look at how these checkpoints work. So there are many other protein complexes that are involved in regulating the cell cycle, and these are going to be involved in 4 cell cycle checkpoints. These are going to be critical points in the cell cycle that are regulated, that are going to stop the progression of the cycle until a go signal is received. So regulatory molecules at each of these checkpoints are going to determine whether a cell is going to go ahead and proceed or it's going to stay in its current phase. So if these regulatory molecules are defective, the checkpoint may fail. And this is going to be the cause of lots of different kinds of tumors, a failure of these checkpoints during the cell cycle. So let's take a look at what these checkpoints are, where they occur, and what they do. So let's first look at the G1 checkpoint. So this seems to be the most important checkpoint in the whole cell cycle. Generally, once a cell passes the G1 checkpoint, it will usually complete the cycle all the way through mitosis. So it's going to pass this checkpoint if the cell size is adequate, if it's big enough to be able to divide, if there are sufficient nutrients available for growth of the 2 daughter cells, if social signals are present. So social signals are things like the growth factors that are coming from other nearby cells. And it's going to check to make sure that the DNA is undamaged. If it's damaged, it will hold the cell in G1 until the damage can be repaired. If it's determined that it can't be repaired, then it will go into an apoptosis cycle and the cell will be destroyed. So mature cells that are not going to divide are going to go at this point into G0 and be in this resting phase, this non-dividing phase. Otherwise, if it passes this checkpoint, it's going to go on through synthesis into the second gap, and there's going to be another checkpoint in the second gap. So in the second checkpoint, it's going to check to make sure that all of the chromosomes have been replicated successfully without damage to the DNA. Replication is a point at which it's very likely to have damage to the DNA. And it's also going to check to make sure that there is sufficient activated MPF available to proceed into mitosis. So if all those things check out, it's going to proceed into mitosis. These 2 arrows represent the 2 daughter cells. In mitosis, there are going to be 2 more checkpoints. One is going to be during metaphase to check whether the chromosomes have attached to the spindle apparatus. So make sure that each sister chromatid is attached to one kinetochore microtubule. And then a second one is going to be during anaphase, checking that the chromosomes have properly segregated, that you're getting one going toward each pole, and that each daughter chromosome is going to get the correct complement, the full complement of all the genetic material. So external factors that influence cell division include specific growth factors. So growth factors are released by certain cells and stimulate other cells to divide. So this is mostly through that paracrine signaling pathway, cells causing nearby cells to go into cell division. In density-dependent inhibition, crowded cells are going to stop dividing. So most cells also exhibit something called anchorage dependence. To divide, they must be attached to a substratum. So if these conditions aren't met, if they're too crowded or if they're not anchored to something, they'll usually not get past these checkpoints and stop the mitosis cycle, stop the cell cycle. So density-dependent inhibition and anchorage dependence check the growth of cells at an optimal density, making sure that you don't get abnormal growth. So if we think about cancer cells, this is, remember, out of control cell division. Cancer cells exhibit neither of these types of regulation of their division. So if we think of these cells growing in culture, the anchorage dependence means that these cells are going to only grow in a single layer. Why? Because they have to be attached to the bottom to be able to divide. Once all the anchorage sites at the bottom are occupied, they're going to have to stop because they're not going to divide if they're not anchored to anything. And then the density dependent inhibition, the cells are going to form just a single layer. If you remove some of those cells, they'll divide until they fill in that gap. So this is how you get a normal mammalian tissue in a single layer of cells. But in cancer cells, the cells just keep growing regardless of whether they're attached to anything and no matter how crowded they are, and that's going to form these abnormal growths known as tumors. So cancerous cells have two types of defects. One can be a defect that activates the proteins required for cell growth when they should not be active. And the second are defects that prevent tumor suppressor genes from shutting down the cell cycle. So those checkpoints that check to make sure that the social signals are appropriate, that the DNA is not damaged, those are going to fail to shut down the cell cycle. So many cancers are thought to arise from cells with defects in the G1 checkpoint. Cells that acquire the ability to divide indefinitely are undergoing a process called transformation. They're transforming into tumor cells. So cancer cells that are not eliminated by the immune system form tumors. So these are masses of abnormal cells within otherwise normal tissue. So there are two types of tumors. One is a benign tumor. So these are non-cancerous and non-invasive. They're just abnormal growths. So here we see an example of a benign tumor. These are the benign tumor cells that are dividing out of control, but they're not going to spread to other parts of the body. These are the normal cells. In pink, we've got a lymphatic vessel coming through and the blood vessel coming through. These benign cells are not entering either the blood vessels or the lymph vessels. Malignant tumors are cancerous and invasive, so they can spread throughout the body via the blood or the lymph and initiate secondary tumors elsewhere in the body, and this is called metastasis. So here we have our malignant tumors in orange, our normal healthy tissue, and these malignant cells are leaking into the blood vessels and into the lymph vessels where they can travel and metastasize into other areas of the body. Now let's think a little bit more about this social control, because this is one of the big problems that cause these out of control growths in cancer. So cells respond to signals from other cells. They divide only when it benefits the whole organism. So this is a coordinated system that creates the right form and function in the organism. So social control is based on growth factors, small proteins that stimulate division in nearby cells. So cells in culture will not grow unless serum, which is the liquid portion of blood, is added to the culture. So the growth factors in the serum are going to allow cells to pass the G1 checkpoint and go ahead and start growing in culture. So cancer cells divide without growth factors. So they may make their own growth factor, or they may convey a growth factor signal without the presence of the growth factor. So they can initiate these pathways on their own without the original signal. So how does this G1 checkpoint work and what goes wrong? So growth factors stimulate the production of a protein called E2F. And E2F protein is going to trigger the expression of the genes required for the synthesis phase. The growth factors are also going to stimulate the production of G1 cyclins. So this is where you start to get the increase in concentration of cyclins. Here we have our growth factor receptor. There's going to be a transduction cascade ending up ultimately in the nucleus where we get this production of the E2F protein and the cyclins. Another protein, RB, is a tumor suppressor. So this is going to regulate, hold back the growth, keep it from going out of control. So it's going to suppress the activity of E2F until we really need to divide. The presence of RB is basically going to keep the cell in the G0 phase and keep it from dividing. So the growth factors are going to help cells pass the G1 checkpoint by doing several things. So first, the growth factors are going to arrive from other cells. They're going to bind with that receptor, cause that transduction cascade that results in the production of cyclin and E2F. So the RB protein is going to be just present in the nucleus, and it's going to bind to E2F, and it's going to inactivate E2F. Meanwhile, the cyclin is going to bind to CDK, the cyclin-dependent kinase, and it's going to be phosphorylated in both the activating site and the inactivating site. So this is going to be inactive at this point. In the next step, the inactivating phosphate is removed, so this MPF then becomes activated. It's got its cyclin, it's got its CDK, and it's got its phosphate in the active site. And what it's going to do is it's going to phosphorylate the RB protein, causing it to release E2F. So when RB is phosphorylated, E2F becomes active, and then the cell can go ahead and enter the S phase. So how does this fail in the case of cancer cells? So in some cancers, the G1 cyclin is overproduced. So we get way too much of the cyclin, and that's going to permanently activate CDK. We're going to have this complex ready to go all the time, and so this is going to be overactive in phosphorylating the RB protein. So if this RB protein is constantly phosphorylated, it can't bind the E2F. E2F is just going to bypass this whole process and allow the cell to go directly into S phase. Another mechanism is if RB is missing or defective. So in either case, RB is not going to bind E2F, and again, E2F is just going to bypass this whole control system and go right into S phase and initiate cell division. So in this web lecture, we've looked at the cell cycle. This is the end of the material for exam 2, and we've seen the different phases of interphase, what the purposes of these gap phases and synthesis phases are. We've looked at the 5 stages of mitosis and the process of cytokinesis. We've looked at how this whole cell cycle is regulated. In the next chapter, we're going to focus on this synthesis phase, and we're going to look at the DNA molecule in great depth and explore how it's the basis for heredity and how this replication occurs during the S phase.